And so in the lab, for the cord ceiling, you'll see something like this. Note that it does not have the epi and hypocaudal. Instead, you'll see the coleoptile, and then once the seedlings grew up a bit more, you'll see the emergence of the leaf here in the red arrow. And the key difference between, well, one of the key difference, one is the number of cotyledon, and the second thing is in eukaryotes, eudicots, the energy for the seed is stored in the cotyledon, and in monocots, it is stored in the endosperm. What happens in eudicot is there is an endosperm to begin with, and then the energy in terms of starch or lipid or sugars gets then transferred into the cotyledon, and then the endosperm sort of goes away. All right, so here is another knowledge check. Go ahead and pause the playback for 2 minutes and think about this. So, name three traits that are different between monocots and eudicots. Okay, so remember from our previous slide, there are several things you can choose, right? I think you're choosing 3 out of 5. You have the number of cotyledons, that is different. You have the venation pattern of the leaves. You have the range of the vasculature. You have the root system. So eudicots have a taproot, monocot have fibrous root. And then you have the flower part. So monocot is multiples of 3 and eudicot is multiples of 4 and 5. You can also put that monocots have energy for the germinating seedlings stored in the endosperm, whereas eudicots have it stored in the cotyledons. So any of those choices are fine. Here is another question. So label the coleoptile in the following photo, and then is this plant monocot or dicot? And what kind of root architecture does it have? Go ahead and pause playback and try to answer these questions. All right, so in this case, coleoptile, you have to go down to the earlier stage of the seedling. So there is a coleoptile. This plant is monocot, as exemplified by the fact we're talking about, coleoptile and not cotyledons or hypocotyls. And because it's a monocot, it will have a fibrous root system. All right. Again, if you have questions, go ahead and drop a message in Slack. You can drop it directly into the lecture materials channel, or if you are still uncomfortable with messaging me directly at this point, we do have, again, the peer-to-peer channel where you can discuss any questions you may have with your classmates. Again, in the student peer-to-peer channel, I will usually not check that channel unless someone at me directly. Okay, so that covers the first part, the main organization and organisms of plant body. And now we're going to move on and talk about biodiversity in plants. So this gets into the difference in the plant body and organs and all the variations you can see. So plants and animals are pretty different in how the growth of the body works. Plants have what is known as modular growth. So we have nodes and internodes, and remember each node and internode is one unit of growth or one module. Animals don't do that. And what that means is if you chop the head off of a plant, it'll do just fine. But if you do the same for an animal, the animal is not going to be fine. And that is because animals, such as humans, we have what's called determinate growth, and the body plan is determined prenatally. So in our case, we have one head, 2 arms, 2 legs. This is usually how it works, and this has been determined before we're ever born. Plants, however, have what's called indeterminate growth, and that means that the body plan is highly malleable throughout the life of an organism, and this is also known as modular growth. This is why you can cut the head of a plant or trim a rose bush, and that will actually make the rose bush bloom more. How is this possible? Well, that's because plants have stem cells, and they do this through the meristem, which is where the population of stem cell is. Because plants have vegetative organs and reproductive organ, you might imagine there's also two corresponding populations of cells. There's the vegetative meristem that gives rise to the vegetative organs, such as roots, stems, and leaves. Then there's the reproductive meristem, which gives rise to reproductive organs, such as the flowers and cones and the associated tissue that can then become seeds. And for stem cells, the general principle is actually the same behind animals and plants because you start off with a stem cell, which then gives rise to daughter cells. A portion of these daughter cells will maintain the stem cell fate, while the rest will become committed to a differentiation lineage and become differentiated cells that have specialized function. Here's a pun that I kept from Professor Lisa Serafini, the previous instructor for this course. And the line of the joke goes this way. Plants are very happy because they have meristems, or in other words, meristems. This is a nice pun to have fall quarter, right? As we're getting closer and closer to Christmas, and some stores start putting out their Christmas products super early. All right, so here's what the meristem does in more detail in a plant. What happens when you chop off the terminal bud or the head of the plant is once that happens, the lateral meristems located in the axillary buds become activated. Normally, they're suppressed by signals coming from the terminal or apical bud. And then the lateral meristem can become the main meristem in the shoot once the terminal bud is gone. So the terminal bud is so important in this case, both in terms of being the primary source of growth and from determining whether or not the lateral meristem is activated or not. And all of this comes down to the meristem at the very tip called the shoot apical meristem. We tend to call it SEM for short because shoot apical meristem is kind of a mouthful. And it's one of the two main vegetative meristems in the plant. And it can also give rise to the floral meristem and lateral meristem. Because remember, you have to have the shoot apical meristem first, that gives rise to the lateral bud or axillary buds. And then the lateral axillary buds is what can then gives rise to the branches, all its associated organs and meristems, as well as become the next stem if the stem is the terminal bud gets chopped off. The floral meristem also derives from this. We'll get into this into more detail. But for now, know that shoot apical meristem is one of the main vegetative meristems. It is protected by these baby leaves called leaf primordia and this dome-like structure. And there are specialized zones in the shoot apical meristem or SEM that gives rise to different lineages of cells and tissue types in the shoot, and we'll go through this in a later lecture. So now I am going to stop sharing this and show a game that is associated with this that you guys can play with in your own time. So this is a game by Japes, and it is how you can really get an idea of a plant as a organism with modular growth because each time you do this, you are clicking to add modules to the plant. And you can see each time you grow a unit of the root, it requires energy in terms of sun and water for resources. But each time you grow a shoot unit, it will also increase the amount of sunlight. Whereas if you add units of the root, it'll increase the amount of water you can absorb per second. So this is a pretty cool thing for you to play around. And the nice thing about this is this game does build into the physics of things so that if you had too much imbalance between the root and the shoot, the plant can actually collapse. So that's something that you can play around with on your own time. But for now, let's go back to the lecture where we have previously discussed the shoot apical meristem. And now we can talk about the root apical meristem because yes, there's meristem populations specific to the root too. And for root, what is really interesting is root is really easy to trace the specific cell lineages because everything is arranged in these perfect columns growing from the root tip upward. There's a lot of different cell types. We're going to cover them in more detail later. But for today's lecture, just remember that for the root, the meristem region is called the root apical meristem, or REM for short. So we have SEM for the shoot, REM for the root. It is the other main vegetative meristem in the plant, and it gets to rise to a lot of initials, which is what we call the meristematic cells in the root that's committed to different lineages of cells. So to summarize, plants differ from most animals in that embryogenesis does not directly generate the tissue and organs of the adult. The body plan is not determined at birth. The entire adult plant morphology is generated by the activities of the apical meristem, the SEM and REM. So in other words, in a typical eudicot, the embryo looks like this. This does not in any way reflect what the adult plant looks like. And you cannot tell how many branches or leaves or root it will have at maturity. All right, moving on to the next section now, where we get into the biodiversity plan, and part of the reason why I think plants are super cool is because we have a lot of variation that's possible, even for the typical body plan between species. You can have variations within the same species, depending on genetics and environment for all of these. So some sources of morphological diversity is genetics. These are all oak leaves. You can see there's a great deal of variation both in the leaf shape and in the shape of the acorns. So here's some terminologies for leaf shape. We can cover this in more detail for the leaf lecture. But in other words, the leaves can be lobe in shape, in which case it's got these large waves, lobe structure. They can have serrated margin. They can have an entire or smooth margin. And oak trees are one of the most famous and well-known trees in California. So next time when you walk through campus or through Land Park, you can take a look around you in your neighborhood and see how many of these leaves have you seen before, and how many of these have you seen on campus. Recall from earlier that we talked about compound leaves. And for compound leaves, each leaf have leaflets, right? Instead of one blade, you have many leaflets, each with their own blade. The central extension from the petiole, where all the leaflets attach to, is called the rachis, and this is the structure that you see with a pinnately compound leaf. You generally don't have a rachis with a palmately compound. And the leaf all come from leaf primordia, which are emergences from the stem. So there are these things that wrap around the dome of the stem. The plural form of leaf primordium is leaf primordia. And how you typically number the leaf is you go from the youngest to the oldest. So here, primordia 1 or P1 is the youngest primordia that just developed. Older than that is P2, and if you zoom out from this, you can see there's also P3, P4, and so on. And one of the reason I mentioned this is to go into the fact that there is a lot of things that affect the shape of the leaf. So that is genetics, the source of leaf diversity. And you can have diversity within the same species. For instance, in this case, there is a gene called trifoliate. And if you mutate this gene, the plants with this mutation will have a different leaf shape in tomato. So here is the P1, P2 that I just mentioned. So these are baby leaves that are just developing. Here's what that looks like in a live plant that has not been sectioned. And the left is if you chop the plant horizontally or in the transverse plane in half. And here's what that looks like in higher magnification using electron microscope. We'll cover this in more detail in a later lecture as well. But for now, I just want to draw everyone's attention to panels I and J in the bottom right corner. You can see in I is what the normal tomato leaf look like, and J is what happens if you mutate the gene trifoliate. You can see now it has 3 leaves, right? That's why it's called trifoliate. And you can see the amount of difference that a single gene can have in the same species on leaf shape. Trifoliate is one gene. There are a lot of genes involved in leaf shape or leaf morphology. And you can have a lot of natural variation within the same species. But keep in mind, there's also variation possible that's within the same plant. And I just want to mention now that we'll talk more about these at a later date. But there is still a lot unknown about how a leaf grows even now. And that's why I wanted to include these slides from recent research articles to show you all the work in plant biology that's still in progress. So in this case, this is a paper published by Martinez et al. In 2020, and it's a collaboration between research team in USA and Japan, because science is not only a community, it's a global community. Now, here are the possibilities of variation that's due to environment. So here is the same species of oak tree and the difference between a leaf that's grown in the shade versus a leaf that's grown in the sun. You can see there's a difference in size of the leaf, the depth of the lobe, so how far these leaves go in, as well as the color of the leaf. And this is what your lab project number 1 will be about. It's about the sort of climate readiness of California native trees. Because California is the state in the US with the most floral diversity, and we have no idea what the effect of climate change is going to be on a lot of these native species. So something that my past students have worked on is they have asked, how will climate change affect leaf morphology and anatomy? How will climate change affect California native trees? And are there traits that are associated with better tolerance? So in the past, and what you guys will be doing is you guys are all going to go out onto campus and find yourself several trees, and you're going to track different traits of those plants or those trees for the quarter. And the final data for that is going to be your lab project. All right. Other aspects of morphological diversity in terms of phenotypic plasticity, in addition to light, we also have temperature. So you can see as the temperature increases and decreases, there tend to be an optimum in terms of leaf size, but there is also variation in leaf shape as well. And in our case, this gets into your project number 2, which is salt and drought tolerance in lettuce. And this question is asked by past students that they have made some progress in answering is how will climate change affect leaf morphology and anatomy? And specifically, the reason that we're continuing this project with you guys is the question the past students have asked is how will climate change affect the leafy crops we eat, such as lettuce? So that's how the lettucing happened. Additional things that can affect a phenotypic plasticity or leaf morphology is that in terms of the plant stages of development that can also have an effect on the leaves. And so this is something called heterophyly. It means that on the same plant, you can have different leaf shapes. And we have seen shade versus sun can change that. But in terms of developmental stage, here on the left, you can see poplars can vary dramatically in terms of their leaf shape depending on which part of the tree they're growing from. So because of the way plants grow, the youngest part of the shoot tends to be at the top, right? And then in terms of Roripa aquatica, so this is kind of a, I guess you call this a water weed, aquarium weed. Whether the leaves are fully submerged or underwater, or whether or not they're growing on the surface, will actually cause a dramatic difference in the shape of the leaf. And something that I thought was super cool that my past student didn't know and also thought was cool when I mentioned this is that typical ivy leaf that you see, which looks a little bit like this middle shape here, right? It has three lobes and kind of a rounded shape. That's actually only the juvenile form of the leaves, and the adult forms of the leaves look super different. They look more like number two of the popular leaf here. In addition to diversity in the leaves, there can be a lot of diversity in other organs as well. So because I was talking about leaves which are a vegetative organ, I want to talk about floral diversity or reproductive organ diversity now. So we all know and love the fact that flowers are very showy, they're very pretty. There's a lot of variation in both their shape and color. And in terms of all the variation you can see in a typical eudicot flower, you can have difference in UV reflection. Depending on whether or not there's temperature differences, you can actually have difference in pigmentation. There's difference in cell type between the tip and the base of the petal in terms of scent, variation in cell shape and texture, and even electrical charge between the different portions of the flower. A practical example of this and reflecting research that's currently underway is the gene cycloidea in the wishbone flower. And this is one gene that regulates the pigment or color deposition of the flower, but specifically in the dorsal side. So this is one organ morphology where the plants have bilateral symmetry, just like we do. And in terms of studying plant morphology, there's a lot of directional vocabulary as well. So in this case, the flower, like us, have a dorsal or backside, a ventral side, which is the front side, and a lateral side, which is going from side to side. And sometimes you also hear this referred to as the sagittal plane. And this is research done by a group in Japan. So something that we can now think about, in addition to the leaf, is what will climate change do to the flowers? Because we know that temperature will have an effect on floral development as well as pigmentation. There's also population genetics in terms of diversity, both for vegetative organ and reproductive organ. In this case, here is an example of floral diversity for population genetics in Calocortus, commonly known as Mariposa lilies. And you can see there's variation in color. And here is a work where the authors or researchers have studied the different phenotype variations and their population, which are grouped and correlated with climate data and their allele frequency, or how frequently each genotype that's behind each phenotype occurs. So one of the things that is required for this class is that you have to go on 2 field trips, and it's a really nice way for you to get out, look at nature, and embrace the diversity that you can see in the plants. So there are 2 field trips offered in this class, considered 2 sections. There is the UC Davis Arboretum. So for people in my section, this is on Saturday, September 20th It's a self-driving field trip. I'll give you guys the location and the GPS coordinate for where we'll meet on UC Davis campus. And we're going to go there and look at all the plant collection in the Arboretum. If you are unable to join me on that day, you can also attend the Arboretum field trip through Professor Christine Grun, and I'll include her contact information as well once we figure out where her date is. The second field trip is a joint field trip between our two sections, and it is on October 25th, also a Saturday. And in this case, we'll be going to Point Reyes National Seashore, and you do not have to drive for that one. We have made arrangements for bus pickup and drop off on campus. And remember, this is one trip only for both sections, and you do have to attend this one. So mark your calendar early, okay? All right. Now for other aspects of morphological diversity, so which is modified organs. So in plants, you can have a lot of modification for the organs. You can think of this like in ocean mammals, like whales or dolphins, right? They look like fishes, but they have vestigial organs or vestigial hind limbs, which are modification of their hind limbs. And you can have similar modification of organs in plants as well, whether in terms of vestigial organs or organs that's been modified to serve another function. So here in this case, we're looking at a cut open onion, and it is a case where we have modification of the leaf. So instead of flat leaves we see in the trees, in this case, the leaves are modified so they're fleshy and they can store a lot of energy. The stem is actually compressed and this tiny thing over here. So the internodes do not elongate. So all the nodes are compressed together in this bottom. And so this is why we have terms like typical, right? Typical eudicot. Because there is a lot of untypical or atypical plant bodies that we see in nature as well. And sometimes this can make it quite tricky for you to identify whether something is a stem or a root. So one of the key identifying features in identifying whether something is a shoot or root is that only stems have nodes and internodes. So in this case, if you trace each leaf all the way to the bottom and look at this in higher magnification or better quality photo, you can see each leaf is attached to this thing at the base. And remember where each leaf attached, that's a node. So once you have nodes, that means there's probably internode in between, even if they might be compressed and tiny. And if you have nodes, that means you're looking at the stem. So here is another knowledge check. This is a lettuce, not lettuce, sorry, lettuce in my brain. This is a celery. And here is what you normally call the stem of the celery, but it's actually a stem. Which organ is this, do you think? And how can you tell? Go ahead and pause the playback, and I'll tell you the answer in 2 minutes. All right, so for celery, if you ever chop open the celery down the middle, you'll find out that celery is actually very similar to the onion in the case that it has actually compressed internodes. So the part that we call the stalk or the stem of the celery that we eat is actually part of the leaf. You're eating the petioles of the leaf. And the reason you can tell is you had to trace where all the growth is coming from. So again, if you break off all the stem of the celery, you notice all of them is attached here at the base. And that is where the stem is, because stems have nodes where all the side branches and leaves grow from. So now for another question to test you again. Which organ is this? This is a ginger. And how can you tell? Go ahead and pause the playback. All right, so as some of you might have guessed, there are these things, these lines, which are actually the nodes in the ginger, because the ginger is an underground stem, also called a rhizome. So again, once you have nodes and internodes, they can come in all kinds of shape and appearances. But once you have nodes and internodes, you're looking at the stem. Last question for this section. Which plant organ is this and how can you tell? If you ever seen these in the Asian market, they're usually labeled in English as lotus roots. Possibly duck. As you might have guessed from my verbal hints, this is also a stem, an underground stem. And the skinny part of each of this is actually a node. And the part that we eat, the wider part, is the internode. So if you have any questions about any of these parts, remember to leave me a message. Make sure you have this clear because remember, knowledge check is a source of the question banks for the exam. So some of these will show up on your exams. So make sure you can make sense of how to tell apart a stem from a root. So my fun challenge for you guys is to go through your fridge and identify which plant organ you're actually eating. If you have roommates, this is a chance to troll your roommates with plant biology. But you can see now, again, I emphasize this is why we keep using words like usually and typical, because there's a lot of unusual and atypical things that happen. And so there's usually a couple of tests that you can use to address the atypical body plan to try to identify what is what and what you're looking at. And one of the most reliable ones, and the ones I want everyone to learn for this class, is to check for nodes and internodes. And just to, again, emphasize how weird plants can get, here is the Brazilian grape tree. You have the fruits growing directly on the trunk. It looks like that. You can have the dragon fruit tree. Try to figure out what is the stem and the leaf on this thing. And you can see here is what the fruit looks like. It's growing at the end of these green structures. There's the ghost plant. Everything you see here is reproductive. Where are the vegetative organs, right? Here's the monocarpher. Here's a reproductive structure where there's these, I guess you call them sort of prickly looking circles attached at the bottom of the leaf. We're going to actually cover ferns in more detail later, and we have a special fern lab that tends to be everyone's favorite lab. So we'll answer that question later. Okay, moving on to the final section of today's lecture, we're going to look at selection in plants, both natural selection and artificial selection. So everyone has probably heard about Darwin and natural selection before, but just to remind everyone, natural selection has four main postulates that must be true in order for it to occur. Number one is that members of population often have variations in their genetic traits. So there is diversity that can be inherited, such as, for instance, the flower color variation we saw earlier within this population. The second thing that must be true is that all species can produce more offspring that their environment can support, and so many offspring will fail to survive and reproduce. In other words, there is competition for survival. The third postulate is that some individuals will have traits that do better than others in that environment. In terms of insects, it can be color of the insect that can help them disguise themselves in the environment. Or in terms of plants, it can be disease resistance, right? So here is tomatoes and some tomatoes are more susceptible to a kind of fungal disease called verticillium wilt. And you can see here are two different species of the fungus, and there are some plants are obviously doing better than others, right? So if the plant is dying from this disease, it's not going to flower and give rise to seeds. And that brings us to the fourth postulate, which is that these individuals that are more fit for that condition will have more offspring and they'll pass down that trait, whether it's disease resistance or color. As a result, over time, the population should accumulate more favorable traits. So again, those are four traits or four postulates for natural selection. And so this really gets into the concept of biological fitness, which is the ability to survive and produce viable or fertile offspring. It also gets into the concept of adaptation, which is referring to traits that can increase the fitness or the ability to produce offspring of an organism in a particular environment. And the adaptation is accumulation of traits as a result of natural selection. In other words, natural selection is kind of like an editing process of the genetic frequency within a population. And the source material of what natural selection acts on is mutation. Mutation gives rise to differences in genetics, which the natural selection can select on. So one of the examples that I can give here, this is a review paper published by a group in Netherlands, is salt tolerance in plant. Because when you have drought, you have to water the ground for the crops to grow. And then the salt ions in the water builds up in the soil, and the soil becomes saltic and salty, and the plants will die from the salt. And you can have salt tolerance as a trait for survival because salt stress is a really complex process. The plants can have variation in their ability to sense the salt stress, in their ability to sense throughout the plant that there is stress and to act on it. And then the plant can have adaptive response in terms of the growth and development that can then come with tolerance and survival. And it is a never-ending game of survival, essentially. So one question I want you guys to take a moment to think about right now is because natural selection is something that's going on for millions of years already, right? Why haven't they evolved to be perfectly adapted to their environment? And the hint to the answer to this question is think about climate change. And for those of you who have paid some attention to all the news with climate change, as well to natural history, which you might have had in a previous class before, is that environment is constantly changing, right? We had ice age, we had increasing temperature now. We have periods where there has been a lot of rain, and we have had periods and areas where there has been a lot of drought. So that's why it's a never-ending game because the environment is changing. There's disease that come and go. And so the selection criteria from natural selection is always changing as well. So an example of this, one of the most famous example is the Galapagos Island finches. And the research question that was asked in this classic experiment is, did natural selection on ground finches occur when the environment changed? The hypothesis posed by scientists is that if there is a drought, there should be a change in the beak characteristics because of the availability of food sources and food type will change, right? So the beak shape would change because birds with beak that's better adapted to eat this new, or I should say, the food source that still survive throughout the drought are going to do better because they're going to be able to eat more. And so if they do better, then they can lay more eggs and give rise to more offspring. The null hypothesis. So for an experiment, we always have a hypothesis and a null hypothesis. The null hypothesis is always that there is no difference. And in this case, the no difference means that there should be no change in beak characteristic in response to a drought. And to run this experiment, the protocol is to go and weigh and measure all the birds in the population before and after the drought. I don't know if they use undergrads for this, but I have used undergrads in my field study before, and like we all go out in the field and measure things. It can be fun and also kind of frustrating, and you can get an idea for that in our Arboretum field trip and also in your lab project number one, where you go out and measure trees on campus to see whether or not you like it or not. If it turns out you really like running out and measuring things, then one of your future career choices might to go and become a field biologist. And here is what the result of the study looks like. So here is a measurement of the finches in 1976 before the drought. And here is the number of finches in the years after the drought. And on the x-axis is the size of the beak. And you can see with drought, there is a shift such that there is more finches with bigger beaks after the drought, suggesting that corresponding to the change in the type of food source that is available. In this case, I believe what happened was due to the drought, there is more availability of a certain type of seeds compared to before because the other plants have died. And to eat these seeds, you need bigger and stronger beaks. So you can see there is an increase in bigger beaks. And here's what that looks like. And again, because environment is constantly changes, there is another drought that occurs, so they can repeat this experiment, right, to see whether or not this thing can be validated. And in science, we always want to validate things as much as possible. And by this time in 2004, because remember, the previous experiment is in 1976, it's been a while, right? So now in 2004, there has been a pretty well-established population of these crown finch with large beaks at this point. And so you gradually have separation of the population from the small beak and the large beak. And similar process can happen in plants as well. So in I don't want to say Western diet because there's so many countries and cultures that can be considered Western at this point. Let's say in a typical uh, Caucasian American diet, you guys are eating about 8 species total. And what happened is that the 8 species just all look very different because you selected to overgrow certain organs of one species. So in this case, kale, Brussels sprout, cabbage, broccoli, kohlrabi, and also cauliflower, which is not here, they all came from the same species. It's all wild mustard. And this is artificial selection. So man-made selection, where basically we selected in kale for plants that have bigger leaves. For Brussels sprouts, we select for the axillary or lateral buds. For cabbage and you're selecting for terminal bud. For cauliflower, you're selecting for large stem shoot apical meristem. For broccoli, you're selecting for large floral meristem. And for kohlrabi, you're selecting for large stems. So these, like the finches, eventually became separate lines or populations, but originally they all came from one population. So both natural selection and artificial selection, if you do it long enough, variations that originally were occurring within the one population can gradually develop into several populations of plants. Another thing, or something that's part actually natural selection, part artificial selection, is bananas. So I call this the story of Captain America's banana. And this is based on the principle of resistance to stress. Plants undergo a lot of stress. It can be abiotic stress, which is referring to drought and heat and so forth. Or it can be biotic stress, or stress caused by other organisms, such as disease, pests, and pathogens. The reason why this is Captain America's banana is that when Captain America was born, most of the banana that was eaten in US is the gross Mitchell cultivar. And then 1950s, Panama's disease happened, which is Panama's disease is also known as fusarium wilt. It's another fungal disease. And basically everything we eat now is a Cavendish banana. So the Captain America who has woken up after his hibernation will find that the bananas will taste and look different because there was Panama's disease that wiped out entire lion bananas. And remember, survival is a never-ending game, right? It turns out that we had a new outbreak of Panama's disease around 2010. So the fight is ongoing. Researchers are still working on it. Here's a news article on BBC about it. And you guys can read this on your own time. But just know that selection is ongoing. Artificial selection is something that we use extensively in plant breeding. So there's a student-run xenia breeding program at UC Davis done through the Student Collaborative Organic Plant Breeding Education. So there's a lot of undergraduate and graduate students who run this entire show. And in this case, people are selecting for new colors, new and unique petal shapes, better stem size and flower size to make cut flowers, right, in a nice bouquet. And what the students are doing is they're crossing existing flowers, which is where you have the variation in genetics, to try to get offsprings with preferred trait. And then they have all the resulting flowers line up and they select for the ones they want, and those are the only seeds that they keep. So this is how you select for it. So now, when we're getting near to our last knowledge check, and this covers another type of question you'll see in the exam, which are the fill-in-the-blank questions. In this case, please fill in the blank for the 4 postulates that must be met for natural selection to occur. Go ahead and pause the playback for now. All right, so the 4 postulates are: 1, members in population must have genetic diversity or genetic variation, or you can say traits that are different, that are heritable. So the key words there are variation that can be inherited. Number 2 is that there must be more individuals that can survive in that environment. In other words, there is competition of survival. Number 3 is some individuals must have heritable traits that makes them more fit or be able to survive better in that environment. And number 4 is that these individuals will have more offsprings that carry that trait. You can just say more offsprings. Again, if you have questions about any of these, make sure you ask, all right? Okay, so slightly more detail about lab project 2, which is the lettuce salt tolerance project, because lettuce and salad green is one of the top commodity crops growing in California. And as you guys probably have heard, we have just a little bit of a drought problem in California, which means we have very dry soil and a lot of salt that's building up in the soil from all the irrigation we're doing to grow these crops. And this project gets into artificial selection for stress tolerance in lettuce and other salad grains. And breeding for tolerance, it will be faster if we know what traits to select for, right? So if you can find a trait that can be seen when the plant is super young, like 2 to 3 weeks old, so you don't have to wait as long and grow up all these plants. You can just grow up these few plants that have these traits and just wait for their seeds. So it makes it faster and it saves money in terms of the resources it takes to grow these plants. So something that my past students have tested is the leaf area to volume ratio. They use weight as a proxy measurement for volume. They have tested for greenness as approximation of chlorophyll content, and they have tested survival. So to be tested by you for the setup this week and next week is root growth. Because what we saw in March this year is there might be association with bigger roots with better tolerance. But you guys are also going to collect some more leaf phenotype as well for the leaf lab. So now the final part of today's lecture is to explain what do I mean by the weekly lecture homework assignment. So your submission for this assignment is going to make up 25% of the exam questions because what you guys will be doing is to the slide deck link I posted on Canvas to Google Slides. Each of you every week is going to add 2 slides. On slide number 1, you're going to make up an exam question based on the course material covered the previous week. And on the second slide, you're going to provide the answer to that question. And then for the exam, for 25% of the points, I'm going to randomly pick questions from you as a slide deck. So again, you determine what goes on your exam. You guys feel free to coordinate among your classmates to decide who's asking what question about what subject, okay? And I will go through the slides every week when I'm grading to make corrections and to make sure the answers are correct. So you get 2 points, but for the answer, you're only going to get your answer point if you provide the correct answer. And what the slide will look like is this. So again, make sure that you put your name, top left corner, the lecture number or the date of lecture that your question is based on. So if your classmate had a question when they're looking through the slide deck, they can go back to the course material of that day and find the answer themselves. And then in the middle of the slide, you can write your question. This will be your slide number 1. And your slide number 2 will be exactly the same, except instead of posting or writing your question in the middle of the slide, you're going to write your answer in the middle of the slide. All right, and that concludes our first lecture for the synchronous section. Again, if you have any questions, make sure to leave me a message, and I will see you guys in lab.